We're happy to welcome Bohdan Schneider, who's a structural biologist and head of the Laboratory of Biomolecular Recognition at the Institute of Biotechnology of the Czech Academy of Science. He studies interactions between proteins and nucleic acids, and his work on annotation and validation of nucleic acid structures and geometry is the focus of his talk today. So, Bohdan, there you go.
Hello. Hello. Thank you for invitation to be part of this nice meeting. So I'm Bohdan Schneider. I work in Czech Republic near Prague at a big institute which is called or building or consortium which is called BioSef, and my institute is called Institute of Biotechnology. I'll walk you through this website that's called nutco.mos.org, and it concentrates on analysis of nucleic acid geometries. Okay, I cannot move. Okay, good. So this is why we work on nucleic acid tools, because we want to make analysis, all these things, annotation, refinement, validation, modeling of nucleic acids more reliable. Why more reliable? I will mention a little later, but in general, because it's not very reliable now. So before I start talking more specifically about the website, I have to tell you what are our tools, or what we actually developed, and how we do the analysis. We analyze nucleic acid structures at relatively local level, at the level of dinucleotides. Why dinucleotides? Because this is the smallest level that makes sense, and the largest level cannot be, or largest fragment cannot be analyzed for still lack of data at such detail. It can be analyzed obviously, but with not at this level of detail as we need it. And we analyze dinucleotide geometry in torsional space. This is a chemical diagram, or if you like it in 3D. So we analyze backbone torsion angles basically between one sugar and the other sugar, with phosphate in the middle, and then we include orientations of both bases that are involved in dinucleotide. So these chi angles, and then we have three additional parameters that are two distances, N distances, nitrogen distances, and carbon sugar carbon distance, and pseudotorsion in between. We derived, using this fragment, we derived 96 classes we called conformational classes we called NTC, dinucleotide class. This is the paper where you can find details how we did it. So 96 classes, what does it mean? It means that each class is a relatively homogeneous conformer, but it still is an ensemble of structures. It's not one average structure. This is relatively important to understand and accept this concept that the each NTC class is actually a distribution, certain distribution of conformers. Then each NTC class has its name, and there are 96 of them, and then some classes, NTC classes, can be grouped together to build so-called structural alphabet that we call Kana. This structural alphabet actually helps some symbolic analysis of the structures, and I'll tell you later how we used it. Okay, So now let's move to the website. First, when you go to the website, you can upload either your own structure or structure which is already in PDB. Here I show some pictures about 5v3i PDB deposited RNA structure. At the simplest level, we do some very easy to intuitive annotation of the structures. You see the picture on the left side with some graphical representation. I will explain more in a minute. And then on the right side, you see a table that has basic characterizations of each building block we work with, which means dinucleotide. You have some name of a dinucleotide, then you have this Kana letter here. You see, it's not actually a letter, it's 3 letters, but it's a Kana symbol, and then NTC symbol, and then 2 numbers. One is so-called confound, which is a torsional measure of agreement with our standards, and then RMSD is normal RMSD in Cartesian space. Green means good overlap, orange, red is worse and worse. I will talk more about this. Now back to the picture, what the symbols are or colors. So here the basic picture tells you very intuitively, this is overall A form, which is not so surprising in RNA because it's well known that it prefers A form. It's represented here by these pink pyramids. But you can very easily turn it or simplify the picture by turning off some features of the picture and highlight only some features of the annotation, and you can immediately see what I call here hot regions of the structure, and you can obviously go into larger detail, greater detail, and highlight really some area of your interest, which is here these two dinucleotides. You can get very detailed information on torsion distribution, and at this summary tab you get information, detailed information about the step. Yeah. You can get very important information what other dinucleotides would be possible at this location, which we call similar here, and you get this diagram that is scalable. And here you see that this assignment to OP11, what we call OP11, open conformer number 11, is quite unique, that other conformers are quite high, because this is Euclidean distance in torsional space. So this is really far, 120 or 100 degrees, and also in RMSD in angstroms, it is quite far. Yeah, even this OP11 is not really perfectly adjusted, or it's not really close to our class OP11, but we still accept it as OP11. Okay, so so this would be say a relatively simple level of annotation, but you can go deeper by combining these NTC classes and this Kana structural alphabet, because this structural alphabet allows you to basically turn 3D structure into symbols, and you can then you don't analyze relatively complex 3D objects, but you analyze one-dimensional sequence. Basically, you turn 3D comparison or analysis into 1D analysis. And this is just without much explanation, 2 examples of our results. In one paper, in a recent paper where we published this newest version of NTC, we analyzed tetraloop motifs, and this is one example of some class of tetraloop motifs where this Z-like structure is followed by 2 open structures, and it forms geometrically very well defined loop. And then this is completely different example. This is example of on the right side, completely different example of DNA duplex wrapped around histone core particle that is not shown here. It will be in the middle. This Tetramer Dimers. And what we discovered actually is that the band of DNA in these complexes is realized by periodic appearance of so-called B2 form. Yeah, this is highlighted by this light blue color. It's not perfectly periodic in any single structure, but when we did overall analysis of all available non-redundant set of the complexes, the periodicity of B2 form was quite obvious. It is in detail. I don't have time really to explain how we did it, but it's quite, I think, convincing, or there's no doubt that this is the case. How DNA bends in nucleosome core particle. This is the paper where we published this. So let's move to more, say, crystallography-oriented topic, which would be refinement. We assigned, because the website very easily assigns any structure, so we decided to assign all nucleic acid containing structures, including NMR, including EM, cryo-EM, including obviously crystal structures, and they contain about 7 million dinucleotides. But what is really bad news is that about 30% we cannot assign to any NTC class we define. At the same time, we know that we are not missing any large NTC class. We maybe, and we almost certainly are missing some classes, but they can contribute by 1% to maybe 2% to the overall. But we are certainly not missing even, say, 20%. Yeah, of that's not. We have good reasons, good statistical reasons why this is not possible, but partially good news is that even when we cannot classify, geometries are still not far. These non-classified geometries are still not too far from what we would call well-behaving NTC class. Yeah, about 50% of these 30%, so about 15% could be refined. So here we are at the Andrea Torrent's talk about errors. We could probably refine and remediate a relatively large part of the PDB. It's not simple, but we have approached some pipeline. We are trying to build some pipeline that would allow this. And I will try to show you the idea about the pipeline. We will stick to this 5V3I structure, and there is a dinucleotide U695A696. It's missing here, sorry. Uracil adenine, it's highlighted here. And it's highlighted here. We don't assign it, but I know that RMSD this dinucleotide is very close to NTC class we call OP12. So we don't formally, for some reasons that we don't have time to explain, we don't assign it. So this conf in torsional space is 0, so it's not assigned, but we know that RMSD is close. And we know it's close to something we call OP12. So if you go to our website, with your structure, you identify a place where you would you feel you could do some improvement. You go to tab, which is called which is called settings. You have to turn on so-called restrain mode. It's originally red. It will turn green. And then you go back to this summary table, and for the selected dinucleotide, you push right. It gives you this yes mark as green, and then in tab download, you can download.
Sorry, but just to say that you have 5 minutes left.
Oh, that's great, because
And that's including the time for questions. So
okay, great. So you can download restraints for FMAC. You can commands for MMB. I'll explain a little bit what it is. Restraints for Phoenix. It's not as simple as I'm telling you, but in principle it works, and we would be very happy to. First, we know we have to make the organization of the website better, but that's always true. But we have to streamline this refinement pipeline. But to do it, we would really appreciate if people use it, and if people tell us what's wrong and what they don't like and how they would like to modify it. So we really need volunteers to use it. And then, in principle, both Phoenix and RefMac restraints are available. This macromolecule model builder MMB, we collaborate with Sam Flores. It's not refinement, it's model building, but this is also very important for low resolution structures, basically. So that's it. Thank you for your time, and I don't know if there are any questions.
Thank you very much for that really interesting talk, and it's good to know that there's an effort being made on the side of nucleic acids and making sure that our geometries are correct. There were I think there are a couple of questions on Slack. So Pietro had asked if there was an equivalent of a Ramachandran plot for nucleic acids, but I think Jiri has replied. But if you want to comment on that.
No, because it's really multidimensional case. Nucleic acids are, and I'm sorry to say that, some protein people are sometimes angry, but nucleic acids are very complicated, and we think more complicated conformationally than proteins, and you cannot really reduce it to 2 dimensions. That's actually error that people sometimes make, and they talk about correlations between 2 particle torsions. It might be true in some context, but it's not true in general, and it only leads to catastrophes. Yeah.
So Jiri had explained that this is a 12-dimension problem. Yeah. And Sebastian Vetten was asking, can you comment why this large number of degrees of freedom can be reduced to mere 100 dinucleotide classes?
I don't know how, but we believe that this is a relatively objective process. There are some classes, some NTC classes could be divided further because it's a result of clustering. Yeah. So clustering is always to some degree or to some level a subjective process. At some moment you say enough. Yeah. So yes, some large groups could still divide, but it wouldn't make any factual difference. Yeah. Yeah. I don't know if it's a good answer, but It is possible. These about 100 classes is, we believe, good number.
I'm sure you can expand the discussion on Slack later. That's one of the reasons why we're trying to get everyone to keep the conversations on Slack, because obviously after the Zoom and even after the meeting, it will still be there. So conversations can continue there. We have time for a couple of quick questions. One from Mark asking, do you think that the problem with the ribosome 5v3i is the resolution, that it is at 3.3 angstrom around the base pair separation?
Yeah, we don't, we don't judge people. We are trying to help to really develop something that will help people to refine. It's actually very good structure, but there are some areas where certainly are issues, but these issues can be remedied. That's the point. Yeah. And that's one direction where we are going. And I haven't talked about correlations. We have also correlations with electron density. So we know where the geometry is close to our geometries, and also electron density is somehow reasonable. Yeah, so it's not only geometry what we work with, but I didn't have time to really show everything.
One last question from Haim Rosenberg, asking if your web server can take into account Hoogston base pairing.
Well, we don't actually consider base pairing. This is single strand analysis. This is the building block. Okay, so this is single strand DNA.
Yeah.
And we don't explicitly discuss base pairing. Base pairing is under development, so we will correlate annotation and then modeling of backbone and base pairing at the same time. But so far we analyze only the backbone.
Thank you very much, Bohdan, and thank you to all the speakers in this first part of the session. I'm conscious that everyone should have we're trying to have a 10-minute break. This is a really long session and we have another keynote. So thank you very much. Unfortunately, I'm not going to be able to be here for the second part of the session. I'll catch up on the videos later. So I'll hand over to Colin and Lucrecia, who will share the next sessions. And we just a reminder that if you haven't had your question answered because you've put it on the chat or in the Q&A, check the Slack channels, because the speakers might have gone there and answered the questions. We are trying to copy all of them there, so they will be answered later. And I think we're back at 20 past, unless Rob tells me, or Mel or Svetlana tell me otherwise. But thank you very much. And I look forward to the rest of the talks and the rest of the sessions.